Hello and welcome to this teaching material on genetic analysis of residual feed intake. I'd like to welcome you to the research farm at Aarhus University called Danish Cattle Research Center. This is a relatively new barn with room for 250 cows, all Holsteins, and as you can see we have in intake boxes to measure feed intake on a daily basis per animal at an individual level. Each of these boxes is allocated to two cows per box, and then you can have different research setups in order to have different feedings for the cows that can go to each each box. Here you can see you have this cow or these two cows here. They are in the blue. In a blue research setup, and this one here is in a yellow setup. So you can make individual measures of feed intake. So the ear tag is red, and when the cow takes out her head, this bar goes up so that this is the only cow that can enter. We have 250 cows. They are milked in a herringbone milking parlor, and there's room for different kind of experiments in the research facilities. Also, we have this machine over here. This is what you call the green feed equipment that is used for measuring methane, also at an individual level. So the cow can walk in here, it gets a little treat, and when it gets the treat, it is also recording methane emission during the visit. So that is summed up to daily feed intake. So my name is Jan Lassen, and I work as a senior research manager at Viking Genetics, and I'm involved in the GenTORE project. And now I'd like to introduce the person that we're going to talk to you about the research that she is doing. This is Rasmus. Rasmus, can you make an introduction of yourself?
Yeah, sure. My name is Rasmus Stefansen, and I'm a PhD student here at Aarhus University. And I work on the GenTORE project with feed efficiency in work package form.
Okay. Can you say a little bit more about how do you define research efficiency?
Well, if we take a look at these cows, you can see what is crucial is that we have these individual measured feed intake for each cow, so we know what they eat, and from their milk production and maintenance requirement, we can calculate what we expect them to eat, and you could say the difference between that is their efficiency. And then the most favorable is that a cow eats less than we expect from her.
Okay, so essentially, if you have a negative value, it is good.
Yep.
Okay. Can you say a little bit more about the data that you are using in this project here? It comes from here, but also other places.
Yeah, so we use the feed intake data here from DKC Forum, but also data from around the world, so from France, the Netherlands and Canada, for example.
Okay, and so Holstein cows, are they just alike around the world?
Probably not 100%. Of course, there's differences because we are using different genetics around the world, but that is also some of the things we'll cover, how genetic different are they, and then also try to estimate breeding values to see what is the difference, for example, between these two cows in their efficiency.
Okay, let's try and walk down the corridor here, and then we can have a little chat on how we can do this analysis. So, efficiency, is that the same trait throughout lactation?
Probably not, because we are working with cows, which is mammals, and so they have mobilization and deposition of adipose tissue and muscles. So you could say there's a huge difference on lactation stage. At least that is our expectation.
And how can you handle that in your analysis?
Well, we can handle that by using quite advanced models called random regression models. So we allow that that animals can, from a genetic point of view, be different throughout lactation. So meaning that, for example, if we have the cow standing there, if she's efficient in early lactation, well, she might not be the most efficient in mid or late lactation.
Okay, so that all sounds very, very nice, but how does this influence the farmers? How can they get use of the data that you have here?
Well,
So what comes out of these analyses here? You get breeding values, I guess, for efficiency, or?
yeah, we get the breeding values for efficiency, and also, especially when we go into genomic breeding values, then we can use these cows to predict the genomic breeding values of other cows.
Okay. Welcome back. So now we are here at another location in the research farm. What is this that you see here, Rasmus?
Well, here's the milking facilities of the research barn. So we have a milking piler here, where at each milking we are measuring the kilograms of milk.
Yes.
And then actually beneath the piler we can take the milk samples from the milkings, where, for example, we can measure fat and protein content, lactose and fatty acids, and you could imagine.
Yeah. So that's also kind of essential, because that's the key energy sink for a cow in the efficiency measuring.
That's where she uses a lot of energy.
Yes. Then down here, what do you see here?
Then we have the running weight. So you could say we get a measurement of the body weight at each milking when she goes out of the milking piler, and that's also very important for accounting for the maintenance requirements, which is around 30% for a modern dairy cow.
So these are the two key measurements that you have. What about body condition score?
Well, every second week, the staff, educated staff, will go through all the cows, and score the body condition of each individual cow. So you could see that takes a lot of labor to do that.
Okay, good. Thank you very much, Rasmus. That's it for now for this presentation. I hope you have enjoyed what we have been telling you, and then we might see you at another stage.